Thank you, Professor. Sterilization is a procedure that is done to eliminate microorganisms from a specific surface through various means, such as exposure to high temperature. There are two main objectives of this experiment. First, to develop a thorough understanding and knowledge of aseptic technique and culture transfer procedures. Second, to learn how to safely transfer cultures of microorganisms from one container to another without contaminating the cultures or exposing ourselves to them. An important technique used in this experiment is aseptic technique. Aseptic technique is required to transfer a pure culture from one vessel to another. Aseptic technique ensures that all prudent precautions to prevent contamination of the culture and spread of microorganisms into the environment are taken. To accomplish aseptic technique, the work area must be cleansed with disinfectant to reduce the number of potential contaminants. The transfer instruments are sterilized. For example, the transfer loop is sterilized by heating with a Bunsen burner before and after transferring. The work is accomplished quickly and efficiently to minimize the time of exposure. The normal steps for transferring a culture from one vessel to another. First, the transfer loop was flamed. Second, the mouths of the culture tubes were opened and flamed briefly. Third, some of the culture growth were picked up by the sterilized loop and transferred to the fresh medium. The fourth step is the mouth of the culture vessel was flamed and resealed. The last step is the inoculating loop was reflamed. Aseptic technique for Petri plate. Inoculating loop must always be sterilized by flaming before it is used to touch any culture material. The closure cap was removed and the tube slip was briefly flamed before the sterile loop was inserted. The inoculating tube was inserted into the culture tube and the loop full of culture were removed. The culture materials was kept covered with their respective lids when transfer was not being done. Materials used in this experiment were: procedure. The single colony of E. coli was transferred aseptically into culture broth, Petri plates, and slant agar using inoculating loop. The single colony of E. coli was tapped into the step agar using aseptic technique. The Petri plates, slant agar and step agar were incubated at 37 degrees Celsius within 24 hours. The growth of bacteria were observed and described.